SAT-1 is, the SAT is the Sumogen AIDS vaccine 001 trial. And this is human clinical trial data. And then I'll talk to you about our new strategy for therapeutic vaccines. So first, let me just go through some introductory slides for those who are not working with HIV vaccines. Now we know that there has been about 70 million people have been infected with HIV since 1981, and there are about 35 million people are living with HIV infection, and there are about 6,000 new infections per day. And this is HIV, and this retrovirus. And those of you who were here this morning, we heard about the structure of the GP120, and that exists as a trimer. And then we have the dimer of the viral RNA, and we have all the proteins. Now the virus has genome of about 10,000 bases, and that codes for about 9 different genes, and there are about 15 or 16 different proteins. So when we talk about HIV, and we talk about this about 10,000 base, the positive stranded RNA virus. Let me just review for you what has been tried in the past. The very first human clinical trial was performed by VaxGen. It's known as AIDSVAX trial and was concluded in 2003. They used recombinant GP120, and that was a monomer. And that did not help to protect the humans from HIV infection. So 5.7% versus 5.8%, and there was no difference. And then we know that in 2007 we had the so-called STEP trial by Merck, and actually the vaccinated people had a higher rate of infection than the control groups due to the pre-existing antibody against AD5. Then in 2009, we were very excited to see this result of the Pasteur, Sanofi Pasteur vaccine known as RV144 trial, and we had 31% efficacy. This was based on canary pox carrying GAC gene, POL gene, and M genes, and then boosted with recombinant GP120. And the 31% efficacy was the best seen so far. And then the 2013, last year, USNIAID, National Institute of Allergy and Infectious Diseases, their HVTN 505 trial is based on DNA vaccine followed with AD5 boost, again had higher rate of infection with the vaccine group than the control groups. So that was discontinued. So these are the basic trials of the human clinical trials in the last almost 2 decades. Now, in addition to these 4 different human clinical trials, now there are ongoing trials right now, and there are about 20 phase 1 human clinical trials, and about 3 phase 1, 2, and 2 phase 2 trials going on. And these include the DNA vaccines, recombinant adenovirus vaccines other than adenovirus 5, and also recombinant poxviruses such as canary pox, MVA, and fowlpox, and also protein-based vaccines and combination of above. Maybe there are some more trials going on. However, it is very interesting to see that no one is using one strategy that is used to develop the vaccine against so many different viral diseases. That is the inactivated whole virus strategy. When Jonas Salk was still alive, and he was advocating to get this killed whole virus vaccine against HIV. But there was a problem with this strategy because no one knew how to produce large quantities, and also it's too dangerous to produce. And by the way, there was a company called, I think it's Reimmune, and they made a vaccine on the basis of the killed whole virus, but somehow they deleted GP120. And that was most important. antigen, supposed to be. So that company is closed. Now we try to make this whole virus strategy. Now this is conventional strategy. However, we modify the HIV by such a way that we can make it less dangerous and produce large quantities. Now let me tell you how I do it. And we just make this avirulent virus by modifying the genes of HIV. We take clade B, which is prototype virus in North America and Europe, and we make it such a way that it's avirulent and replication efficient. Now, how do we do it? Now, we know that the NEF gene is responsible for pathogenesis. That studies from SIV work as well as HIV work, and also cohorts of HIV-infected individuals, NEF gene is responsible for pathogenesis. So we just try to delete that. We can delete it. And also NEF gene is responsible for downregulation of cytokines, such as CD4, MHC class 1 and 2, CD3, and also CD28. And also enhances the virus infectivity. So if you delete NEF, obviously you're making virus production even worse than natural settings. So how do we make this? We can overcome this problem by switching the signal peptide of the glycoproteins. Now this is the clade B glycoprotein signal peptide, and this one we have published almost 2 decades ago, and it has unusually high numbers of positively charged amino acids. And there are, see, these are arginine, lysine, histidine, arginine. And now the signal peptide has a single or at the most a double positively charged amino acids. So what we did is we tried to look at the efficiency of the signal peptide for the processing of the glycoproteins. Now this is actually published in 1994, exactly 20 years ago. And here we have natural signal peptide of the HIV. And now we have pulse chase experiment, as you can see, that most of the proteins remain cell-associated after 8 hours of synthesis. Now, if you delete the natural signal peptide, of course, it does not glycosylate. It doesn't go through the glycosylation pathway, and the protein is degraded really fast. However, look at this one. When you switch the natural signal peptide with a melatonin signal peptide, we can produce a large quantities of glycosylated protein, which is now found in the extracellular fluid. That means that it is secreted very efficiently. And so does the interleukin-3 signal peptide, does the same thing. So what we want to do is we want to put the exogenous signal peptides into HIV. So what we did is we made the so-called the triple mutant, that is the NEF deleted, and then this signal peptide was replaced. Now, because of the overlapping reading frame between the signal peptide of the envelope and then VPU protein, we also lose VPU, so that we are losing the NEF negative, VPU negative signal peptide replaced HIV. That replicates very well. Actually, it replicates much better than wild-type virus. So we have the triple mutant, which is NEF negative, VPU negative, signal peptide gene replaced HIV-1, and it is produced in human T lymphocytes, but it is inactivated by 2 methods. One is a chemical, it's called 2,2' dithiodipyridine, which actually inhibits the zinc finger formation, and also so we can burn it by gamma irradiation. Make sure that there's not a single protein, single virus is alive. I mean, single virus alive virus is one too many. And FDA asked us to demonstrate that. And then we used the montanide as adjuvant. And FDA asked us to reduce the human DNA down to 10 nanograms per dose. So we went through all these painstaking experiments. We have to go through some 230 different tests in the GLP facility in order to get the permission from FDA to do the human clinical trials of this. And this cost a lot. And so we had a master cell bank, we had master virus bank, PD engineering run bank, and the GMP clinical lot and toxicology studies. And with this one, we went through and human clinical trial, phase 1 human clinical trials. That is to study that the safety and immune responses. Now, FDA suggested that we should use the HIV positive individuals because they have nothing to lose. So we recruited HIV positive volunteers. And trial objective, primary objective was, of course, evaluation of the safety. And then the second objective was evaluation of immune responses. Now, the study population was HIV-1 infected adults, men and women, with the ART. And then number of patients that we have recruited was 33. And the vaccination was a single intramuscular injection with 0.25 ml containing 100 micrograms of the viral proteins and plus 0.25 ml of montanide. And then our design was to have at least minimum of there are 4 arms. The first arm is killed whole virus with 9 people, and placebo was 3. Killed whole virus with adjuvant, 9 people, and adjuvant alone with 3. So these were the plan, and then we follow up with 12 months. This was performed in LA and 3 hospitals, and we decoded this one, unblinded the results by April this year, and we discovered that there was 13 people in killed whole virus alone, and 4 people in placebo control, and there was 12 people in killed whole virus with montanide, which is adjuvant, and then 4 people was in placebo control with montanide alone. And I'm not going to show you all the data, all the details, but I'll just summarize what we found. There was no significant adverse events of those vaccinated with all the vaccines. And also, there's no viral load increase in vaccine subject, and no abnormalities were found in hematology or biochemistry laboratory evaluations. In addition, we found that there is the antibody. Remember that these are the HIV positive individuals who carry the memory cells against HIV, so that they do have a threshold of antibodies. And now when we immunize with this killed whole virus, it will stimulate to make more antibodies. So these are the groups we have. The arm A is killed whole virus alone. 41% showed anti-P24 antibody, elevation of anti-P24 antibody. 25% of the people showed the P17 antibody elevation, and then the 25% shows also elevation of antibody against GP120. And this is a control group, and 1 out of 3 people showed that, the elevation of antibody slightly. And this is the real vaccine, and we have 72% of the people showed the elevated antibody against P24, 72% shows elevated antibody against P17, and then 45% shows elevated antibody against GP120, and 9% shows the elevated antibody against P41. And then this is the control group, shows the marginal level of antibody productions. So this is a summary of immune responses. The 4 P24 antibodies, arm C subjects showed as much as 128-fold increase, and arm A shows 16-fold increase. Okay. And the P17 antibodies, arm C, which is a real vaccine, shows as much as 500-fold increase, and then arm A shows 8-fold increase. And there's GP120 antibody shows that about up to 16-fold increase, and GP41 shows 2 to 4-fold increase. So killed whole genetically modified HIV-1, which is we know as SAB1, with adjuvant montanide, boost adaptive immune responses. This result actually forecasts the phase 2 clinical trials because this is a really good immunogen. Now, we hear so much about the native structure of the HIV proteins. We hear so much about the trimers of GP120 and also native structure of all the proteins of HIV. Now, we are presenting here as the native structure of entire virus, which is killed. So summary is genetically modified and killed whole HIV-1 vaccine, SAP-001, is completely safe in use in human. And SAP-1 boosts antibody productions in HIV-positive subjects. And also adaptive immune responses result in phase 1 human clinical trial, forecasts possible outcome of the phase 2 human clinical trials. So this is for the prophylactic vaccine. So we are now preparing for the phase 2, and it is very difficult to find CROs, is a contracted research organizations producing these vaccines because this has to be produced in the BSL-3 GMP facilities. There are many companies can produce that for us, even if we pay millions of dollars. But anyway, we found a couple, and they will produce. So this is good for phase 1, and hopefully that this is good immunogen. Hopefully that this may show us some protection. We don't know. And presently, we are processing, we just got finally the antisera from those patients. And we want to see whether or not these people also produce broadly neutralizing antibodies. And that's the next experiment that we want to do. Now, what about those people who are living with HIV infections? 35 million people. Now, can we do anything about it? We see drug-resistant viruses and all kinds of problems associated with these chronically infected people. Now, of course, there is no therapeutic vaccines as yet available to treat the virus-infected individuals. But can we do it? Now, in order to do that, I think we have to do 2 immune responses. Number 1 is antibody-dependent cellular cytotoxicity. That is the humoral immune response. And then, of course, the CTL response. And in order to have these, we have to have a system to produce viral proteins inside of the cells. I'm not going to go into the details of those mechanisms. You all know better than I. And now what we can do is, in order to produce HIV-specific viral antigens in infected individuals, we have to have the vector system which can infect human cells. Now we chose to use raptovirus system. And the prototype of raptovirus is known as a vesicular stomatitis virus. And the VSV, it has a very wide host range, including human. And it has a very mild human infections, and also no pre-existing antibodies in human. And M gene mutants are avirulent. Now I'll go into that later. And the 2 antigenically distinct VSVs are available. The one antibody against the one serotype will not neutralize the another serotype, so that we can use one serotype with HIV, carrying HIV genes, and as a primary immunization, and then we can boost with second serotype, which does not cross-react, cross-neutralize, as a boost immunization for the maximum boost effects. And then we can produce this virus very high titer. We can get easily 1 to 4 times 10 to the 9 PFUs per ml in 8 hours of infection. And also, we can use all kinds of cell lines because this has a very wide host range. And it can accommodate as much as 6,000 bases. So we can put lots of HIV genes into it. And the level of expression of the inserted gene is very high, and you will see that. So we are using the priming vaccine vector with the Indiana serotype. This is Indiana isolate, priming, and then boosting with New Jersey, and which will carry the same set of HIV genes. So no matter how good the vector system is, if it is neurotoxic or if it is detrimental for the vaccinees, we cannot use it. So we had to make sure that we can make a safer vaccine vector. So in order to do that, we found that the conversion of the methionine to the arginine at the position 51 of matrix protein makes virus very safe. In addition, we learned from the literature that we can make this virus as temperature sensitive. And Enflammont from France, some 40 years ago, isolated a mutant of TS mutant, temperature sensitive mutant, and that was the assembly defective mutant, and we found that there were 2 mutations in it. And those are the important because glycine to glutamic acid change and the leucine to the phenylalanine change makes this virus temperature sensitive. And so it's very mild and it is temperature sensitive. In addition, we also generated the mutants of the VSV New Jersey with the same mutations. Methionine to arginine change at the positions 48 and 51 makes this virus. We published this one about 5 years ago. And this virus also is very, very safe. So we are using these vectors and to carry HIV genes. Now, in order to show that, but first of all, we have to mutate these negative surrender RNA viruses and then recover these. But the recovery is done by so-called reverse genetic. This is a well-known technology now that we can introduce any mutations in RNA viruses and we can recover that by using reverse genetics. It has T7 RNA polymerase promoter and also hepatitis delta virus ribozyme sequences, so that the replication of transcription of this one by T7 RNA polymerase can give you exact ends of the RNA, and that can be replicated by providing RNA polymerase complex, so that this will produce those mutant viruses. It's known as reverse genetics. So we can produce that. And then we look at whether or not these viruses will produce exactly the same amount of proteins at the permissive and semi-permissive temperatures. Although assembly of the virus is very poor at non-permissive temperature or the semi-permissive temperature, which is body temperature, 37 degrees, the level of protein expression, just look at this, is identical at the 37 degrees as well as 31 degrees. And the same with New Jersey, same level of the virus protein synthesis. So although it is a temperature sensitive, now then we look at how safe is it. So we can isolate these viruses, inject into the intracranial inoculation by bird hole. So we can buy those animals from the animal suppliers, that they sell these with bird hole, and we can inject as little as 5 microliters. When we inject 1,000 virus particles into the mouse brain, they all die with a wild-type virus in 4 days. But those mutant virus, we can put 1 million, which is 1,000-fold higher, it does not touch. And same with the VSV in New Jersey, it's less neurotoxic. However, we see the paralysis of those animals, although they don't die. However, those mutants that we have created, they are all safe. So we have safe 2 vectors, VSV Indiana and VSV New Jersey. And using these 2 vectors, we are inserting now the structural genes of HIV. So we are using GAC, POL, and EMV, and put it into the VSV. Now, what we are doing is we are putting these genes, GAC, POL, and EMV, between the G gene and L gene of VSV. And also, in addition to GAC, and we put the T-cell epitopes as a chimera. So it's chimeric proteins are produced. So GAC protein with the chimeric proteins, and then RT in the presence and absence of the protease genes, and also entire envelope genes. And then we can look at the expression of this, and you can produce the proteins at the high level, and this is the processed RTs between the virus-infected cells, and this is GP120. So priming with the VSV Indiana carrying these HIV genes and boosting with VSV New Jersey carrying the same set of genes, and we have immunized mice. And we look at, I will show you just the couple of examples of these immune responses. This is the gamma interferon Elispot test, and showing that this is VSV, this single peptide stimulated target cells, and this is N peptide of the VSV. It shows you that all different settings. Now VSV alone shows you very nice the CTL response, and then this is the VSV carrying GAG gene, shows you GAG specific the CTLs, and then this is in the M specific CTLs is very, very high. This is percent of the total T cells. And now we used the three different peptides of the pol, shows you the positive response. And then this is a mixture of the GAG, pol, and M, but the peptide itself is M. Again, shows you very nice the immune response, it's CTL response. And this one is a mixture of all different peptides. In addition to CTL response, we looked at humoral immune response, and we see that this is the antibody ELISA against the P55 GAG, and we see that the antibody against the GAG is produced, as well as when you do the ELISA against GP120, and we can see that GP120 is expressed antibody is expressed very well. So why then do we get the humoral immune responses from virus-infected the animals? And because we looked at that, as you know, that it has been reported, we did that too, that if you delete the protease gene and express only P55 GAG, and it produces virus-like particles, and they secrete from virus-infected cells, as well as GP120 is secreted from virus-infected cells. Therefore, we do get not only the CTL response, but we get the humoral immune response. And that is shown the next slide, that when you look at those virus-infected cells, the VLPs, virus-like particles, these are the GAG particles are produced. And then when you look at the GP120 in extracellular, actually the fluid of the infected cells, you can see also GP120. Therefore, we get the humoral immune responses as well as the cellular immunity. And so we'd like to now try this system to get into the non-human primates, and then eventually getting into the HIV positive individuals, and see if we can reduce virus load, as well as we can make those people viral free. I mean, at the moment, of course, the HAART treatment can prolong patients' lives for a long time now, but there are always the issue of the drug-resistant mutants. And now, can we get rid of the virus chronically infected cells in the body, such as the macrophages or T lymphocytes? And can we do that? And then perhaps we can help those people who are chronically infected with HIV. And of course, we need money to do this. And so the conclusion of my talk is that HIV, GAG, pol, and M genes were inserted into the genetically modified VSV Indiana and VSV New Jersey vectors. And then HIV1, GAG, pol, and M were expressed efficiently and processed properly. And priming with the VSV Indiana HIV virus and boosting with the VSV New Jersey HIV virus induces high levels of CTLs, as well as the antibodies against the GAG and M. So conclusion is that the genetically modified dual serotype VSV system carrying HIV structural genes could be very useful for immunotherapy. Hopefully it will work. And also, by the way, this platform technology is now available and for you to try out. And anybody who is interested in using this system, I'll be happy to collaborate with you. And of course, this work is supported by the Canadian Institute of Health Research, National National Research Council of Canada and the Health Research, and also the company who is sponsoring our research is Sumogen and Canada. And so we do all our work. By the way, next year, we are going to host, in fact, I'm going to host the American Society of Virology meeting on our campus. This is the Western campus. And those of you who are interested in attending ASV, please come and meet me again. Thank you very much. I'll answer any questions, maybe 2 questions before we take a break. Yes?
Yeah, excellent talk. What I'm trying to ask is the main problem today with the HIV people is the reservoir virus, which doesn't get eliminated. So is something going to be done with this system, whether it's going to take care of that or?
We do know that there are so many different clades and so many different mutants available and circulating. Now, when we look at the human epitopes for the CTLs, there are many, many, many epitopes in different the viral antigens. So that even if there are variety of different the epitopes, the clades available, I think there are many cross-reacting T-cell epitopes in many different proteins of HIV. So hopefully that this one prototype will take care of the many different epitopes of HIV infections. Yes, Dr. Spear.
I think I'm not sure whether you're mistaken or not, in saying that nobody's really tried killed virus vaccines for HIV. If I remember correctly, way back 30 years ago, there was a guy in West Africa, I think his name, I don't believe his name was Rivers, but it was something like that, and he tried to do it, and he was basically drummed out of the field because he was actually injecting a killed virus, which meant that this was a formaldehyde inactivated HIV. And nobody was sure that he'd really got 100% inactivation, and you never are, because you get particulate formation, and formaldehyde doesn't inactivate everything anyway. So he got drummed out of the field. So he had to go. Then there was a guy called in Nigeria, I don't know whether you know his name, is Abalaka. And he and I sort of spent basically a weekend together composing a paper for his work, which was involved in inactivating HIV. I think he was using some kind of heat inactivation, and he was basically injecting this into the soldiers in Nigeria, and he asked me if I wanted to go as well, which I politely declined, and we went on from that. And then there's another guy in Thailand, I think, I've forgotten his name, it's something like Bandra Nayaka, and he's also had a go at using an inactivated HIV. So, you know, there are precedents in the literature for what you're saying, but I do think that the presentation you've given today has been wonderful, and I think it is possibly a beacon to a much brighter future for this bloody virus.
Well, thank you. I don't have as many contacts as you have around the world, and I do know some of those episodes, and it did not go further, and only actually officially launched the clinical trial was by the Reimmune, and that was killed HIV without GP120 on it. And I don't know why. And then Dr. Sok had a patent actually on that, and it did not work. Of course, we do need GP120. And when we look at, actually, I did not show you any characterization of these viruses that we produce, but the ratio between GP120 and P24 are identical as a wild-type virus shows after purification of the virus. We use FPLC to purify the virus.
Thank you.
Yes.
Well, trying to. He didn't do it.
Well, he formed that Reimmune. Actually, he was the advisor for the Reimmune, and then they made those company, but did not work. No. Yes.
How does the spike stand up to the Montanide? Did you look after you put it in the Montanide adjuvant?
Montanide alone did not show any side effects. And I heard that somebody had some experience with some side effects with Montanide alone, but we did not have any side effects in our cohorts.
I mean, on association of the 120, staying with the spike, not the adjuvant itself, but the impact on the spike structure? I
don't know. I cannot answer that question. Yes.